Welcome to Club EvMed, Virtual Evolutionary Medicine Conversations. Nice to have these slides up so I can remember exactly what the full title is. This is our, roughly twice a month meeting that happens where we talk about the latest in evolutionary medicine and get together and get to hear one another's perspectives. I mean, first of all, hear about some new perspective, but then share our ideas and perspectives on that. So thank you all very much for joining. My name is Charlie Nunn. I'm at Duke University in evolutionary anthropology and the Duke Global Health Institute, and I run TRISEM, the Triangle Center for Evolutionary Medicine. And I want to point out that this seminar series is actually run by a bunch of different evolutionary medicine centers. We have an organizing committee, and I think I have all the centers represented here in the chat. And it's also organized by the International Society for Evolution Medicine and Public Health. And I'll try to paste that in. And this is a great time to renew your membership if your membership has lapsed, or to join the society, which is really the Society for Evolutionary Medicine. And please submit articles to the Society's journal, Evolution, Medicine, and Public Health. And very importantly, I want to call attention to the annual conference that will be in Kiel, Germany, July 28th to the 31st. And that registration is going to open earlier than usual before the holidays. And we really hope that you'll join us there. Should be a fantastic conference. We've got keynote speakers starting to get lined up, and you'll hear more about that as we go. So for these Club of Meds, we often like to have a book tour of sorts, where an author of a recent book gets a chance to talk about their book, get some feedback and insights from others, and have a good conversation about the book. And so we're really happy today to have one of those book conversations. And I'm going to turn things over to Randy Nesse, who organized this, and he'll introduce the speaker and also a discussant. Go ahead, Randy.
Thanks so much, Charlie, and welcome everybody. I'm thrilled that this has all come together. When I saw Lawrence's book, I knew I had to read it immediately. I follow his work every time something gets published. I read it immediately, and he's one of the more creative, or most creative, evolutionary geneticists of our time, and going along with David Hay, who's agreed to provide commentary. So I think we're going to have great fun. Introductions are really brief for Club EvMeds. Lawrence is at Bath in England. He studied with Bill Hamilton, and you can see that influence throughout his work. David is at Harvard, and I love on his webpage, he starts off by forthrightly saying, "I'm a theorist," and that theory has done us so much good, and I encourage all of you to read his work if you haven't read it already. I think our topic today is especially timely. The book, The Paradox of the Organism, is coming out in just 2 weeks from Harvard, and I think the whole topic of genomic and intergenomic conflict associated with the whole issue of small population sizes and vulnerability to mutations is going to be very central for evolutionary medicine in the next decade. And here we get a head start. Lawrence, over to you for a quick introduction.
Well, thank you ever so much, and thank you for the invite. So first off, apologies if what I'm about to tell you appears a bit simple to you, but the book is intended for I guess for British 17-year-olds upwards. But I think I was told it works very well with American medical students as well. But it's basically an attempt to put into some sort of context some recent thinking about why we are not very good organisms. So, but to put that into context, you could try doing this. Put the word evolution into Google image search, and I can tell you what you get back, because what comes back is nearly always some version of this, the so-called march of progress. And it's many people's understanding of the evolutionary progress. But evolutionary biologists, for the most part, don't like it because of its biases. And those include a bias towards the supposition that evolution is somehow progressive, that it's directional, and possibly worst of all, that somehow humans are sort of the fairy on top of the Christmas tree, I suppose you could say, at this time of year, or the end point of the process of evolution. And as with all things, Shakespeare does it a lot better than anybody else. So here's Hamlet saying, "What a piece of work is man, the paragon of animals," the very best that the animal world or nature has to offer. But there is good reason to suppose that this is in fact how the evolutionary process works, that it is indeed progressive, because our canonical neo-Darwinian model envisages one type better suited to its environment outcompeting the resident. So present-day species should be better adapted than prior ones, survival of the fittest, all of that. And when we teach our first years, we naturally point to something like the pepper moth, a beautiful example of, in many regards, progressive evolution. When the trees went white, the pepper moths went white. When the trees went black, the pepper moths went black. But I would also argue that when we look at the world through genetic or DNA lenses, things look rather different, and that it doesn't seem to be all that progressive. And I'm going to argue that, in fact, to a first approximation, in fact, it isn't. So there's a whole series of mysteries and challenges that we have to face. So the first that came out, again, 2003, we've now sort of honed it down a bit, is about 1.2% of our genome actually codes for protein, the canonical job of DNA. What does the rest of it do? Well, there's about 8 times as much, which is dead retrovirus, so-called endogenous retroviruses, and the vast bulk is old transposable elements, line 1, alu, etc., etc. And again, we've got more pseudogene than we've got gene. And there's a little bit of non-coding RNA, which is no doubt functional, and so on and so forth, but it's only about 1% doing the canonical job. And this is not inevitable. So what you see here is intergene span on the x-axis, log average intron size on the y-axis, and there's us fairly close to the top. And the striking thing is these two variables, although they're independent variables to measure bloating, these two variables are highly correlated across species. So every data point there is a genome. And, you know, we're large and bloated. You go down to yeast, then nearly all of the yeast genome is, as far as we can see, functional. The average intergene distance is tiny, the introns are tiny, and there aren't many of them either. Another I would point to, and this is much more David's home territory, might say, is I think it is quite remarkable how hard it is for humans to have a healthy child. And this captures, I think, many interrelated issues that I think to any evolutionary biologist should be having you scratching your head. So the first of these is that it turns out that most fertilized human eggs are not viable. So when we look at frozen eggs or embryos, we see typically 40 to 60% of them die because they've simply got the wrong number of chromosomes. So we know about aneuploids in terms of rare Down syndrome cases. So 98+% of births have the right number of chromosomes, the canonical 46. But when we look at the embryos and the early fertilized eggs, that's more like 50%. So the others just die. And we estimate that for every child that's actually born, there are another 2 others that died somewhere along the route, a lot of those being chromosomal errors, early chromosomal errors. And it becomes even more striking when you realize this is a particularly human, or as I'll show you, mammalian problem, because frogs and fish don't seem to have this problem. But there are many other problems that I would argue that are in addition to this. So there are some recent evidence that if you look at chromosomally at individual cells within an embryo, 90% of them have some issue. We've got human-specific diseases like pre-eclampsia, the second global killer of mothers around the planet, and it affects 5% of human pregnancies, going to 10% for older mothers. And we have remarkably high maternal childbirth mortality rates. Currently in the Western world, it's 0.01%, but ancestrally it's more like 1% or a 10% lifetime risk. And if you look up the Forbes list of the world's most risky jobs, none come even close to pregnancy and childbirth. They're not even close. But in addition to that, we have a remarkably high load of genetic diseases. And so a rare genetic disease is defined as, well, in the EU, less than 1 in 2,000. The US definition is approximately the same. So that's the incidence rate. And it turns out that about 5% of humans have a rare genetic disease. Why is that? Well, we sort of know the answer to that one. It's because we've got a stupendously high mutation rate. So what you see here is some parent offspring sequencing data that we put together. On the x-axis, how big is your genome in total amount of coding sequence, and on the y-axis, what is your mutation rate? And as you can see, there's no obviously clear trend there. It's a sort of V-shaped distribution, but who's got the highest? That's data point 8. That's us. We've got a very high mutation rate at 2 times 10 to the minus 8 per base per generation, and that gives us a high mutational load and a lot of mutations as a consequence. So I'd say all of these things challenge, they don't disprove, but they certainly challenge the march of progress view of the world. And then the question is, of course, what is it that we are missing? And here you can take two views. One is that our genetics is perfect, we just don't understand it yet. And this I tend to find is the assumption of most molecular biologists. There's a function for everything, we just have to find it. And the other is that our view of evolution as a perfecting process is somehow wrong. And what I would argue is it's actually the second of these for the most part. But my overall premise for the book is actually quite simple, and it's that if we question the aspects of our biology that don't obviously make sense, this is how I actually define an imperfection. It's not a moral statement, it's just this needs explanation. That's what I mean by an imperfection. We might have to come to a better understanding of the evolutionary process, and in turn then why we have so many medical issues, why 5% of us have rare genetic diseases. And so what I actually argue in the book is that in parallel with the technological advances in genetics, there have indeed been advances in our understanding of the evolutionary process that explain what I would call our rotten genetics. And I would particularly argue that two domains are preeminent. First, as Randy was sort of intimating, the fact that our effective population size is of the order of 8 to 10,000, we evolved in Africa, and that was the population size. That has big consequences according to the nearly neutral theory, and I'll explain a little bit more why I think that's important shortly. And the other is mammals, humans in no different, have flexible resource allocation across the placenta, including, but also including lactation. And in no small part, we have a particularly invasive placenta, and I think these have consequences. And I will look at both in turn. So here's Ota. She developed the nearly neutral theory. An interesting story there. Kimura told her not to do it, apparently. It would be no different from the strictly neutral. And it turns out that it is. And you might notice the credit there, the Royal Swedish Academy of Sciences. So there is no Nobel Prize in evolutionary biology, but there's the Crawford Prize, the Crawford Medal, which Bill Hamilton won, incidentally, but so did Ota. And I would argue that the nearly neutral is probably the most important theoretical advance over the last 50 or so years. But what's remarkable is I don't think as a theory it has penetrated far outside of evolutionary circles. Certainly my molecular biology friends all approach everything as there must be a function for everything. So what does it actually say, and why is it important? Its basic take-home message is that selection is less efficient. Purifying selection will be less efficient when populations are small. So it basically counterposes two forces. So on the one hand, you have drift, whereby we see mutations, slightly dense dust particles that can go up in frequency and they can go down in frequency, and that's drift, that's the chance component. But then there's the selective component, which is what Ota added. The drift component alone is just chimeras. Ota adds to that the weak downward pressure of selection against these mutations as well. And what she finds is that when population size is small, that drift, the selection effect becomes less important and the drift effect becomes more important. And that's really nicely captured in this, what I think is the most incredible of plots. So this is basically Ota's result. What you see on the x-axis then is the effective population size. On the y-axis is the fixation rate. So S equals 0 is the classical neutral case, the strictly neutral case as advocated by Kimura, where the fixation rate is the mutation rate. But what's important about Ota's result is if you consider the figures here below the line, these are weakly deleterious mutations. So S 10 to the minus 6. So this is something that reduces your fitness, but only by 1 in a millionth. So not itself going to cause a genetic disease. What's important about the form of this distribution is that if your population size is small, like us, we're 10 to the 4 or so, then the fixation rate of these is of the order of the mutation rates, are the same as the neutral. But if your population size is large, selection is very much more efficient, and so it will get rid of them. So the net expectation of all this would be if we now ask a question, for example, about a transposable element jumping. It's jumped, it's found rare in a population as a new insert. What do we expect to happen to such events on the average? Well, humans with small population sizes, Ota's theory says selection will be inefficient, chance will be important, and so there'll be an appreciable rate at which these will fix, and so you end up with a large bloated genome. My favorite organism, otherwise yeast, massive population sizes, selection is going to be efficient, chance relatively unimportant, and so you expect a compact live genome. And so we can go back to those genome bloating numbers, put in effective population size where we see them, and what we get is Mike Lynch's result, which we've sort of extended and shown is robust to proper comparative methods. But what you see there is the effective population size on the x-axis and the degree of genome bloating by two measures, the intergenic size and the average intron size. And it suggests that we've got large introns, large bloated genome, simply because selection is an inefficient process. A lot of noise in this data, but the overall trend, I think, is quite clear. Now, where it becomes even more interesting is the extension to this by asking not about, say, a transposable element insertion, but instead asking about mutations that modify the mutation rate. And the same logic will apply, that a mutation that slightly increases your mutation rate can go to fixation if your population size is small, but it's not going to do that because selection is more efficient when your population size is large. And so this extension of the nearly neutral to the drift barrier hypothesis makes a clear prediction, that is that species with large populations with lower mutation rates measured per base pair per generation. So if you remember that plot that I showed you before, which was the V-shaped distribution, we redid this, but now with effective population size on the x-axis, and you see a most delightful trend that species with large effective population sizes have low mutation rates, down to 10 to the minus 11 in some cases, and there's us again right in the top left-hand corner with the highest mutation rate. I don't think it's only effective population size, longevity and what you see there in the whales, they're slightly lower than us. But that's probably hangover to cancer. If you've got a long generation time, there's also selection for a slightly lower mutation rate because you're more likely to get the second hit if you live longer. But the conclusion of all this is if we ask why do we have a high mutation rate which gives us all those rare genetic diseases, then the answer is because we evolved from something with a low effective population size where selection was inefficient. This is not an adaptation. We are not evolving to have a high mutation rate because we need to adapt or anything like that. It's because selection is simply inefficient, is what I would argue the data supports. So what about the second argument? So the second argument I raise is way outside, I would argue, the domain of this weakly deleterious mutations or effectively neutral mutations. We're now in the domain of strong selection. And it's all to do with resource flexibility. So the funny thing about mammals, as David has pointed out multiple times, is that because of the placenta, we have this constant interchange between mother and fetus. And that means that resource allocation is flexible. And this flexibility has rightly been thought to explain many related phenomena. Gestational diabetes with the fetus under selection keep glucose in the maternal blood system, the mother under selection to take it out of the blood system, high blood pressure and imprinting disorders giving extreme birth weights, etc., etc. I don't particularly want to talk about those. I go into those a little bit in the book, but I don't particularly want to talk about those. Rather, I think that there's another effect which I think may be even more important. That's called reproductive compensation. Now, reproductive compensation concerns what happens when an embryo dies. And I think this can explain our aneuploidy problem. But before we go there, let me consider something that doesn't have reproductive compensation, or at least not very much reproductive compensation. And one extreme here, I would say, is the kiwi. So here's the kiwi, and I picked the kiwi because it is, in a sense, at the end of a distribution. So the egg of a kiwi is 25% of its body mass. Now ask the question, what happens if the kiwi egg is in fact inviable for some reason? What's going to happen? Well, the mother has already given all those resources over. Those 25% resources are now gone. They've been handed over. Also, we know, I read the literature on this, and it turns out female kiwis and males will sit on a dead egg. So they waste the time and they waste the resources. So when their embryo is dead, there is no compensation for the death of that embryo. Now contrast that with a mammal. The egg, of course, is initially, and the zygote is initially an absolutely minuscule proportion of the female's body mass. We're not talking 25% to start with or anything like that, just minor fraction. Now, if the embryo isn't then viable, then often the mother doesn't even know there was a conceptus there. And what we know is that most pregnancies, quotes pregnancies with both conceptuses, do not actually make it through to week 6 even. What that actually means is that time and energy can be given away to viable embryos, unlike what we see in the kiwi. And so there is strong reproductive compensation for the death of embryos in mammals. And you can try and calculate this. So I attempted to do a calculation based on survival curves prior to modern neonatal care. And it turns out if you look at a pair of twins growing in utero, versus what happens if one of those twins dies early. If one of the twins dies early, the singleton resulting will then be 40% larger. And if you track that 40% larger through to what their chances of survival are through to age 5 or so, that's a 10% increase in survival chances. So we're massively outside the domain of nearly neutral here. Remember, that was looking at effects that are of the order of 1 in a million, 1 in 100 million, something like that. This is a 10% advantage, so completely different ball game. But what are the consequences of this? Well, I would argue that at first sight this looks like an absolutely perfect system because you save resources that would otherwise have been wasted. But it does come with consequences. So imagine, for example, you have a lethal, embryonic lethal mutation. So we'll just do a simple bit of population genetics. Let's say the lethal version is small a, the wild type is big A. If small a is fully recessive, so only small a embryos die, what that means, and if they do it early enough, that death enables reproductive compensation. And we already saw that. The mother may not even know she was pregnant. She certainly saved a lot of the resources and will save a lot of the time. But then who benefits from this reallocation? Well, from straightforward Mendelian genetics, we know that to make those small a's, dad must have been a heterozygote, mum must have been a heterozygote. So a quarter of the progeny are these lethal embryonic lethals, and a quarter will be heterozygote, sorry, a half will be heterozygotes, and a quarter will be the big A homozygotes. So of the survivors, 2/3 of the survivors have the embryonic recessive lethal allele. By contrast, the capital A allele doesn't see compensation much at all because it's mostly not involved in pregnancies that are associated with this embryonic death. And so what then happens is that this reproductive compensation has the consequence of increasing the frequency of mutations that kill embryos. And R.A. Fisher noticed this, and Ian Hastings up in Liverpool has been doing a lot of work on the consequences of reproductive compensation. But you can see, I'm sure, already that actually this is going to have some interesting consequences because the effects are so large as 10% reproductive advantage. And what I argued quite recently is that I think this can also explain why so many embryos have non-canonical numbers of chromosomes. So here's the logic. So if we think about female meiosis 1, so I would argue that female meiosis 1 in the 8-cell stage, I won't go through to the 8-cell embryonic stage today, but I would argue that female meiosis 1 is the time of greatest jeopardy because here you have maternal and paternal chromosomes segregating, but meiosis 1 in females has the odd property that half the chromosomes are going to a polar body, which is a dead end, it dies, and half are going to go to the egg. So here you have a time of enormous jeopardy for chromosomes. You could have survived all the way through in the female, but if you're going to the polar body, you effectively are not going into the next generation. So just put on your Machiavellian chromosomal hat and think what's the best thing for a chromosome to do. So if a chromosome actually could know that it is going to the polar body, it can't lose. It's got zero fitness in effect. It can't lose by not cooperating. So imagine then a mutant centromere, and I emphasize centromeres here because they have no recombination, so it's always going to be paternal-maternal segregation at the centromere. So what if it were to detach from the Martic spindle when going to the polar body? Well, it's going to win. But the most amazing thing is these are actually known. They detach, in mice it's been shown that centromeres, there are versions of centromeres which if they're going to the polar body conditionally detach. If they're going to the egg, they don't. They hold on to the centromeres, so they detach. But what's the effect of that attachment is it creates a triploid embryo. So effectively what you've got is a chromosome that's going to the polar body. It's got no fitness, and by detaching, it kills the embryo by making it triploid. It wins. Why does it win? is because it's going to induce reproductive compensation. And so the next embryo, all the same embryos within the same womb, so if it's a multi-brood offspring, that same chromosome will on average be in half the progeny, and so those surviving ones win. And if it's like us, it's singleton broods typically, then it's going to be have a half a chance of being in the next progeny. It can't lose in many regards. So in fact, the invasion conditions for such self-fixed centromeres are extraordinarily broad. You just need some good level of reproductive compensation. And if you don't believe me, see the maths in the appendix, as it were. But why should you think this is true? Well, the first and most obvious prediction of this is that aneuploidy in mammals should come about because of meiosis I errors predominantly, and that's well known. So in sperm, the aneuploidy rate is about 1%, maternal meiosis rate is 30 to 70%, there's one report up to 70%, and these nearly all occur in maternal meiosis I. So here's, for example, chromosome 16, where a particular paper looked at 22, and all 22 were maternally derived. Should be associated with centromeres, not exclusively, but largely because centromeres are where there's no recombination, and it's where the spindle attaches, so for those two related reasons. And this seems to be well described. So we know there are aneuploidy effects with age, for example, and this is all due to centromeric cohesion properties. We also know that when the aneuploidies are created, they're because of spindle attachment problems at the kinetochore. And the theory also says that you should expect suppressors of these, and there should be suppressors which are autosomal typically, but somewhere else on the chromosome, and attempting to glue back centromeres to the kinetochore to the spindle, and these are in fact known. There's a BUB1 is a protein which in fact suppresses this particular effect by gluing centromeres to kinetochores. Now, the third prediction I think is the most, in many regards, intriguing one, because what it says is we should be able to predict the rate at which you get aneuploids across the mammals, and it should be approximately a function of the degree of reproductive compensation, approximately constant in mammals, but I think there will be a distinction between the multi-broods and the singletons. But the number of chromosomes is key as a predictor here. So what you see here on the right is, in fact, what the literature says is the rate of aneuploidy. So for pigs, they have 18 chromosomes, and their rate's 14 to 18%. For mice, they have 20 chromosomes, their rate's 20%. Cows, they have 30 chromosomes, and their rate is 30%. The odd one out here is humans. Our chromosome number is 23, but our aneuploidy rate is 40 to 60. A couple of reasons possibly why that is. Could it be an artifact? Much of the analysis comes from IVF embryos. I don't think that's the only thing, though, because if you look at younger mothers with frozen oocytes from cancer, you get similar sort of numbers. But much of the data does come from older mothers, and that's probably quite important. But another effect I would argue is that actually humans are unusual, that we've got an extended period of maternal particular care afterwards, and our brain does still keep on growing, unlike the other primates. And so we might have more reproductive compensation because we've got longer reproductive, care after birth as well. So slightly tilting the balance, as it were. There are, interestingly, two exceptions. One of them, I think, is not a problem at all. That's inbred mice. They don't have a high rate of aneuploidy at all, but I think this is expected. First, it may well be bred out during making of inbred lines. The other is, from evolutionary theoretical points of view, with inbred mice, there is no centromeric asymmetry. And from evolutionary theoretical point of view, there will be no conflict because maternal and paternal centromeres are identical. I don't know which of those is more important. The one I don't understand is fruit flies. They seem to have enigmatically about 1% per chromosome. They probably do have some reproductive compensation, but nothing like as big as mammalian ones. So I'd say that's an open question, and certainly is a problem, I would argue, for my model. However, the nicest evidence, I would say, is not that. It actually comes from a paper which appeared afterwards, looking at a chromosome 2 effect on mice, thing called responder to drive, R2D2, how witty that is. And what this shows is if this particular variant is, as you see here in the top partition of the figure, if it segregates towards the egg, all is well, and you get a euploid egg. If it segregates towards the polar body, it drags it and then releases the spindle is released, but it simply drags the chromosomes and it makes a triploid embryo, and it does exactly what the theory says. If you're going to the polar body, defects. You've got nothing to lose. The next one is probably going to be a fine euploid one because you're heading towards the centrosome, heading towards the oocyte, I should say. I would say there's a number of other unresolved problems, but I raise this just because I want to raise a much more broad view of things. So, as I mentioned in the introduction, one of the striking features is when people go and look at early human embryos and go at the single cell level, turns out that 90%, even of young individuals, 90% of human embryos have chromosome problems in some of the cells. And these are mostly not, because they're young, they're mostly not maternal meiosis 1 errors. Some are, but mostly not. But they're nearly all mosaics for chromosomal imbalances of some variety. Most remarkable paper, this one, really does demand explanation. And my wild speculation stroke conjecture, but I raise this just to broaden the sort of theoretical context here. And while I've shown you an argument that a chromosome heading to the polar body could act in a toxic way towards the egg by making it triploid, there's no reason that is the only way it need to do this. You could have conditional expression of a toxin, for example, or you could have a classic 2 locus distorter, possibly made in nerve cells, acts early in the zygote mitoses unless neutralized, and guess what has the neutralizing factor? It would be the second locus of the 2 locus classic distorter. And it would just simply always pay for such a thing to do it. And we know something similar-ish in mice. So in mice, a T complex is a Martek drive gene, and it kills sperm that don't contain the T complex. But there's no reason it shouldn't kill zygotes that don't contain it as well. So the logic is, I think, exactly the same. So I think this combination of reproductive compensation plus asymmetrical meiosis 1 really leaves mammals and humans vulnerable to all sorts of funny shenanigans, selfish elements acting very early on in embryogenesis and meiosis. So I would say for these broad 2 reasons, we have a very rotten genetic lot, lots of mutations because our mutation rate is high, all sorts of other problems through development. But of course, what's also happened is genomics revolution is giving a medical genetic revolution. And I'd say that is slowly dawning. I think we're actually really seeing it now. And of course, the order comes more or less with this new genetics. We get improved diseases, which get understanding of diseases, which gives improved diagnostics, which improve therapies, and potentially even personal therapies, and now real true genetic cures. So gene therapy has recently been approved for a number of disorders in the UK and is certainly approved in the US as well. And so maybe, although the big brain is our problem because it may well give us ultra high levels of reproductive compensation and probably is associated with small population sizes, it may also be our redemption in terms of genetic medical revolution. So I will leave you with one final thought, which comes from Goethe. And so unlike Shakespeare, I think Goethe actually got it right. And so Goethe said, the man who, with insight enough to admit his, I've obviously inserted genome here, the man with insight enough to admit his genome's limitations comes nearest to perfection. And if it was football, I would say this is most assuredly Germany 1, England, and the behalf of Shakespeare 0. Thank you very much for listening. And have you got any questions?
Thank you so much, Lawrence. What a wonderful, clear summary for anybody who would think that if that can substitute for the book. It doesn't. There's lots more in the book, so don't inhibit yourself. Instead of taking questions quite yet, please put your questions in the chat so that we can keep track of them. And we're going to turn things over to David for a brief commentary, and he and Lawrence might go back and forth a little bit, and then we'll get on to everybody's questions. David, you're on.
Thanks, and thanks, Lawrence, for a great presentation. So I think I'll start very briefly talking what I think are some open areas in nearly neutral theory, and then to one of the loves of my life, talk about pregnancy and early embryogenesis. So in the nearly neutral theory, the important variable is this thing called effective population size. which is sort of a mathematical fix that population geneticists came up to express what the population size would be if it behaved like our simple neutral models do. And it's since that beginning has become one of the most important variables in evolutionary theory, as Lawrence mentions. For me, one of the interesting questions there, so Mike Lynch has talked about the mutation drift barrier. So this is the point at which selection to reduce the recombination rate becomes basically ineffective, and that mutations at repair loci are now don't get any more efficient because they're on the other side of the drift barrier. In some of Mike Lynch's models, this leads eventually to mutational meltdowns because as the DNA repair enzymes become less and less efficient, they become less and less efficient at repairing their own mutations. And in a grim future, this could lead to the eventual death of all organisms that have relatively small effective population sizes. One thing I've been thinking about of late is that an adequate model of what the real population sizes we are looking at? If, and to take another extreme, suppose that a mutation occurs in a cell in the germline and then is immediately eliminated because it's having some cost to cellular fitness, then that has relatively little genetic load, and those sort of and the effective population size is now becoming much larger of the number of cells in the germline or something like that. And so there are issues there I haven't fully figured out myself, but I'm perhaps not as fully pessimistic as Mike Lynch in his most pessimistic moods that we're doomed to mutational meltdown and eventual extinction. To move to look at early human embryos, and Lawrence is absolutely correct. It's remarkable what is happening at that time. Most estimates suggest that the majority of all embryos that are produced are lost early. So the sort of low ball estimate is 50% of human embryos are eliminated, and the high estimates go up above 70% of all human embryos are not making it through. Put that in the perspective, that's saying that most of the natural potential for natural selection in the human life history from age 0 to age 75, 70 years and 10 or whatever our age is, that most of the mortality is actually occurring within those first couple of weeks. And we would expect all sorts of strong selective forces acting in there. And they have been largely not considered by evolutionary biologists. I think that Lawrence is right on looking at the effects of various forms of drive and how they interact with reproductive compensation. I'm uncertain that the rate of aneuploidy from meiosis 1 errors is quite as high as some papers suggest it is. The field is fairly murky. If we look at miscarriages, so they are losses that occur once a pregnancy is established and then the pregnancy is lost. They do show about the majority of miscarriages have various chromosomal aberrations. The question is, are earlier losses of the similar sort? The other thing that we have information on is if you look at early human embryos, as Lawrence pointed out, they have a remarkably high frequency of chromosomal aberrations in some cells, but not others within the embryo. So these are mitotic events rather than meiotic events. So they're not the segregation of the asymmetric meiosis, but something occurring afterwards. They've been a concern in IVF practice, but there are a number of studies that have asked, does it make a difference whether you implant an embryo that's got some aneuploid cells in it versus an embryo that at the prenatal diagnosis doesn't have detectable aneuploidy? And essentially, there seems to be very little difference between whether you put one of the good embryos in or one of the bad embryos. And the suggestion is that there's also a lot of potential for self-correction of embryos at these early stages. So I think I'll just leave it there and to say, yes, we do really, really need to understand better what is happening at the early embryonic stages, because that's maybe where 60–70% of all natural selection in human life history is occurring. So thank you.
Thank you so much, David. Lawrence, I don't know if you
Yeah,
have
if
a
I
very
could,
brief reply, that would be good, but we
how could
really
I have a
want to have
brief
time for
reply to
everybody.
that,
So go ahead.
Randy? Okay, yeah,
It's not possible to ever read the book. Go
read
ahead.
the book. So the any problem, two quick comments, or three. Yes, I absolutely agree that intra-germline competition is going to affect any, and it can also have an interesting effect because we know that some of the competition works the other way. You get mutations in male germline that make the pre-sperm particularly efficient, effectively, and so they cause a lot of male mutation bias. Things like Alpert syndrome is due to mutations that make the sperm particularly efficient, so all Alperts are coming from males to a first approximation. But I'd raise two other points. One is that sex itself and recombination is, of course, a very good means to prevent mutational meltdown. But I actually think for mammals, the bigger one is actually biased gene conversion, because what we do in biased gene conversion is we identify GC-AT mismatches and we repair towards the GC. And given that the mutation bias is GC to AT, for the most part, what you're going to be doing is identifying new mutations and repairing them. So I actually argue in the book that because we've got all these problems, doesn't evolution have something to say about this? And I think, yes, it does. That's why we have sex. That's why we've got biased gene conversion. In terms of the early embryos, the 90% issue I don't think is, it's interesting that it's going on, but I don't think it's necessarily all that profound an issue. So we had a look at, let me tell you about 8-cell embryos. So the 8-cell we think is a particularly important point in time, because this is where the embryo goes from everything being methylated with the transcripts coming from the mother, switching over to demethylation and then remethylation. What we get at that 8-cell stage is massive upregulation of transposable elements, and these then insert. And we had a paper in PLOSB a short while ago where we showed that you get spontaneous line 1 allo insertions at this point, but the cells so affected are detected by DNA damage response, and then they get pushed off to apoptosis, and that's about 20–30% of the cells at about day 5 in the human embryo. And so it's between cell competition that we think is happening in the early embryo. And my guess is that's why many of these single cell chromosomal anomalies are not too important, is because they simply get outcompeted in the rush to become the inner cell mass. So it's really that our filter is who are we allowing through to the inner cell mass versus heading off to apoptosis or indeed pushing off to be placental. As regards the aneuploidy rates, I think there's a really interesting set of stories there. So yes, miscarriages, the miscarriage data says it's about 15% miscarry post recognition week 6, going up to 65% for older mothers. But they partition approximately as half of those are chromosomal, of which half are embryonic aneuploids. Another quarter of all, so half of the genetic ones, are chromosomal issues but post-meiotic. And of the others, about a quarter are simply late implantations, and the system seems to be wired that if the mother doesn't recognize she's pregnant early enough, menstruation simply follows, and that gets rid of a number of late implanting embryos. And the others are immunological intolerance, from what we can see, rather similar, we think, to pre-eclampsia. But if you look at those early miscarriages, they tend to be of the relatively larger chromosomes. But if you look at the very early embryos, the rates are higher for the larger chromosomes. So chromosomes 1, 2, and 3 you see in the very early embryos. By the time you've got through to week 6, those really big ones, if they're triploids, they've gone. They're just too damaging. And of course, why is it that 21 sometimes makes it through the Downs ones? Is because it's a relatively small chromosome. In fact, it's the smallest of the autosomes in gene count, I think. So I think all those things are going on. The actual rate of aneuploidy, as I said, the best data comes from largely farmed animals. So the cow data is quite good because that's all IVF and there's no choice, as it were. But the pig data is quite good. And some of the bird data is okay as well. So birds have a very low rate, whereas the frogs and the fishes with no reproductive compensation have no rate. But the birds, I think, birds will be particularly interesting because some will have a little bit of reproductive compensation if there's a lot of helping at maternal or paternal feeding of the chicks at the nest, such that if the death of a chick allows the survivors to have more resources, there should be a little bit of reproductive compensation there. So I think the scaling of them works approximately with the theory. Whether the absolute numbers do, I think, slightly up in the air, but it does seem to be about 1% per chromosome. But it would be really nice to have really good figures for many of these things. But the birds are so difficult because they've got so many tiny little chromosomes.
There's an extra chapter in your book already developing here. That's and I'd love to hear all of it, Lawrence, but we promised people that this is supposed to be a discussion.
Discussion, absolutely.
And Ned Place has quite a specific short question, so let's have the short question and an answer. Ned, please unmute yourself.
Thank
Go
you.
ahead.
Beautiful talk, Lawrence. Thank you. I was curious as to the population size of chimpanzees and how they compare in terms of the rates of embryonic aneuploidy and loss relative to human.
Any is probably about the same. I couldn't find any data on chimps. There's one paper in other primates, was it a macaque? And it had rates approximately going along with its chromosomal number as well. But that's the only other primate data I know. Nearly all the others, I said, comes from farm animals, where they actually have to look at they do IVF. As a matter of course, that's typically not done with chimpanzees or gorillas, certainly not in the wild.
Let's go next to Cody. I don't see your hand raised, but if you unmute yourself, otherwise we can just ask the question, why do a few trisomies survive through birth and beyond, certain ones and not others?
And I think Lawrence answered this, chromosome size. Yeah.
Chromosome size, yeah.
That makes sense. Thank you.
I still think it's a mystery that 16 and 18 are there.
Yeah.
Edwards and Patow.
Just to jump in on my favorite chromosome 19, which is one of the smallest ones, but is full of pregnancy-related genes, and it doesn't turn up in trisomies. And so the thought there is that it's trisomy per chromosome 19, getting down to the 21 size, is a very early embryonic lethal.
Yeah, incidentally, there's very nice data in mice. No mouse is born with a trisomy, as far as I'm aware, but they do occur naturally. And the time at which the embryo spontaneously aborts is a beautiful linear function of the chromosome size. So ones go early, then it's seconds, then it's thirds, then it's 4, and so on and so forth. So it's chromosome size to a first approximation. A very good first approximation.
Yeah. On trisomy 21 is perhaps best studied, Down syndrome. Even though it makes it through to birth, the majority of trisomy 21 fetuses miscarry.
They do,
so the ones that we see are just a small proportion of the trisomy 21s that are conceived.
The same is true of 18 and 16. And in sub-Saharan Africa, no Down survives past age 5. It's Modern advances in healthcare, you might say.
So Cynthia Bell has a couple of questions related to this. Maybe they've been answered by the discussion. Cynthia, do you want to unmute yourself and say hello? Maybe she's not able to be here anymore.
I'm here. Sorry.
Okay.
I was thinking about people like Pascal Gagnon, who have written about the extensive ways in which mammalian mothers can rid themselves of mistakes and faults during prenatal development. And so I'm wondering if you took your analysis to a larger span of the reproductive period, if you might come up with a different perspective.
Oh, I think that's absolutely true. Yeah. So my actual experimental work on and the theoretical work I do on embryos is either just before the 8-cell stage, very, very early embryos and looking at their fate, or very late pre-eclampsia 20-week onwards. And I think the pre-eclampsia problem is completely different. There, I think it's a problem of improper implantation causing maternal immune rejection of the embryo. So I think it's a completely different story. I don't think it's adaptive in any sense. I don't think there's a selective story to be told about it. It's just a funny and awful consequence of having a very invasive placenta. So yeah, it's I think the in many regards, as far as I see, the least interesting phase of pregnancy is actually the second trimester. There's an awful lot going on in 1. There's an awful lot going on in 3 in terms of preeclampsia, but 2 seems to be really relatively dull. Not that I've ever been pregnant, but
avenue for future work.
Well, not for me, but yes, not experimentally at least.
Okay, on we go. Neil, I'm going to save you a question for the end because it's a practical one about the future. But next, let's go to Abigail. Could you unmute yourself and go ahead with your question?
Hey, Lauren.
Hello, Abigail. How nice to see you again.
Good to see you too. No codon questions today, but I was wondering if you could talk about the pausing in mammalian egg development in meiosis. So there's 2 pauses, right? Do you have any hypotheses about how these pauses interact with your hypotheses?
No, but I think it's really intriguing that it happens. So what I'm actually moving on to just that question and the much more general question of why it is that very early human fetuses have about 6 to 9 million eggs, but at the maximum only about 500 of them ever mature. And why is it that they're held at meiosis 1? It's just there has to be a rhyme and a reason for it. I do not have a good answer for you yet. But watch this space, because that's one of the things I'm trying to work out at the moment. It's such a great question.
Okay.
That's not evidence, by the way, that transposable elements are explaining some of the variation between eggs within the germline, because they have this real burst at the 8-cell stage, and some do make it through and keep on trying to multiply past germline differentiation. And certainly in mice, there's very nice evidence that some of the ones dying are dying because of transposable elements. But that says nothing to why on earth have they been paused at meiosis 1? Why not just wait? And why is meiosis 2 right only after the, in humans, it's right after the sperm has actually penetrated? Why don't you just do it? Make a haploid egg there and then and get it fertilized? But no, they wait. There has to be a very good theory for this. There has to be.
So we have 5 minutes left to talk about the practical implications of all of this. And one is how these things are going to transform our basic understanding of disease. And also, Neil, are you there? Do you want to ask briefly about the practical implications here for genomic treatments?
So I've been following, for example, David Luce's incredible work on a variety of different conditions using a variety of different methods he's elaborated from the basic CRISPR approach. And right now, they're all extremely expensive and hard to imagine on a mass scale being implemented. But let's say in the future that these approaches become much cheaper and more widely applied, what would be the implications for evolution if we're doing a lot, not germline engineering, but somatic engineering?
Well, this actually takes you back to a much broader question, which is what is the relationship between medicine and evolution/population genetics? And as far as I can see, approximately every 20 years, one of the great brains in population genetics is exactly the same thing. So Muller said it in 1920, then I think Haldane said it in 1940s. Bill Hamilton said it in the 1980s. Mike Lynch said it in the 2000s. And what they all said was this, that medicine is effectively keeping harmful mutations in populations. And it should lead to an effectively genomic decay over time. The logic being that what we call purifying selection is, in medical terms, the death of a kid. But because we're doing things to keep kids alive, if the kid's got cancer, we do the right thing. So ethically, the right thing to do is very obvious. But of course, because it's not germline editing, the effect of this is we are keeping those mutations hanging around in the populations. And it doesn't matter whether it's gene therapy or for haemophilia, for example, if it's blood transfusions and we're keeping the people alive and so on and so forth. But yeah, in a sense, medicine by definition is anti-natural selection. If what natural selection mostly does is purifying selection, we are keeping those mutations hanging around in populations.
I'll just make one, it's an interesting response, not a surprising one. I'll just make one other point, which is that I don't think it's been emphasized enough, that definitions for evolution as it's normally conceived of in biology don't necessarily apply well to evolutionary medicine. And my prime example for this is Dawkins's concept of extended phenotype, where he requires that for us to call it an extended phenotype, there has to be selection. But from a medical point of view, in other words, selection on the gene in the organism manipulating the other organism to get benefit from the extended phenotype. In other words, the imposition of a phenotype on a target organism by an originating organism.
So Lawrence and David, you want to say anything brief on that? We have 1 minute left.
I've never thought about things in terms of extended phenotypes, but so no, I have nothing more to add on that, David.
Okay, I'll just have one brief comment. One of the proposals to deal with the accumulation of deleterious mutations is germline gene editing. It's starting to, there are definitely people out there trying to do that. The gene editing that's been done at the moment is somatic. I would just be very cautious. You don't set a person who has no appreciation for poetry to edit William Shakespeare, and we understand very, very little about what the genomic text is doing, and so the chances that an edit will improve. It's in the case of obvious typos, that we've got a functional gene that has got the wrong amino acid, encodes the wrong amino acid. That's relatively easy to correct. The more general thing of the general deterioration of the genome being 5% less efficient or something like that, that's the sort of thing that I just can't see editing technologies dealing with.
No, I agree entirely. So the one attempt to do this was an edit of CCR5 by the Chinese doctor He, and even then, there's a great irony here because he gave his talk at a conference on editing and what have you, on stem cells and embryonic editing, and the talk after his was one on the ethics of it and why you shouldn't do it.
We have to wrap up, but this conversation goes to a future club probably. Adrian Wilson has a book coming out in a couple of months called On the Future of Species. And he's talking specifically about how genome editing will be used willy-nilly to do all kinds of things. It's kind of exciting and very frightening. Charlie, do you want to say anything else to wrap up after this wonderful discussion?
Yeah, just very briefly, thank you, Lawrence. That was fantastic. Congratulations on your book. I'm really looking forward to getting a copy and really appreciate you spending time with us today. David, fantastic to see you. Always appreciate your insights. Randy, thank you for organizing. Jennifer, thank you for your help with organizing everything. I just want to point out our next Club of Med is in 2 weeks. Let me try to get that in the chat. It's Amanda Leah from Vanderbilt. She's going to be talking about a recent paper in Science that they published, adaptations to water stress and pastoralism in the Turkana of Northwest Kenya. It should be a great presentation. We have a couple more lined up already for the spring, and we're going to be filling that out over the next few weeks. So thanks everybody for coming, and hope to see you at the next one.
Thanks, Charlie, and everybody.
Thank you ever so much for the opportunity
Bye-bye.
to speak. And it's so nice to see some Ann Pusey's here. I used to share an office with them.
It's great turnout.